Hi, everyone. My name is Taylor Jorgensen, and I'm a third-year neuroscience PhD student in the lab of Dr. Martha Gillette, co-advised through MBM by Daniano and Justin Rhodes. And the research I'll be sharing with you today focuses on the diurnal variations in hippocampal astrocyte coupling. So one of the major fields of study that my lab is interested in is that of circadian rhythms. And circadian rhythms are biological processes within our body that maintain a cycle of approximately 24 hours. And while they do have this strict cycle, they can be disturbed by environmental changes such as shifts in light. And we know that circadian rhythms in our body are controlled by the SCN, or suprachiasmatic nucleus, which is located in the hypothalamus. And the SCN is known as the master clock of the body, but we see circadian rhythms in other brain areas and all the tissues of our body as well. And so one of those areas that we're interested in is the hippocampus, which is found in the temporal lobe of the brain and is known to be important for memory and learning formation. And we know that the hippocampus includes different specialized cell types, such as place cells and sequence cells, that help to encode different portions of our memory. And it's important to study the hippocampus, as we know that it is implicated in multiple neurological diseases and disorders, such as Alzheimer's and depression. Within the hippocampus, we do in fact see a connection between the rhythms of the SCN and this tissue, as disruptions in SCN function actually interfere with learning that is based in the hippocampus. Some of the core clock genes that control circadian rhythms, such as PER1 and PER2, are found within cells of the hippocampus. And we know that in mice models, if you knock out these genes, we see impaired responses to long-term potentiation, which is a major experimental model for learning and memory. And specifically in our lab, it has been found that we see daily oscillations in both the membrane excitability and redox state in the CA1 region of the hippocampus. And we've actually found this in the dentate gyral region as well, with these regions being opposite of each other. So depending on the time of day, these neurons are more likely to fire than in others. But we're not just interested in the neurons of the hippocampus, but in the glial cells as well. So specifically, I'm interested in the astrocytes, which are a very abundant type of glial cell that are active participators in the tripartite synapse and actually help to ensheath synapses. And among their myriad of functions, just a few are that they regulate the glutamate action in the synapse. They can help to reuptake and release this glutamate. They're known to phagocytose weakened synapses and manage pruning that is actually independent of the microglial pathway. And we know that these astrocytes don't work alone. In fact, they work in network with each other through gap junction coupling. Gap junctions themselves are formed by two connexin hemichannels on adjacent cells that combine together and allow for molecules of up to approximately 1.5 kilodaltons to pass through. And since these channels care more about size than they do about ionic concentration, this allows for these gap junctions to aid in transport of many metabolites and ions, such as glucose, lactate, and importantly, potassium, as they buffer it away after neuronal activity. We see gap junctions and these connexins in many types of cells, neurons, microglia, oligodendrocytes, but specifically within the astrocytes, the two main connexins are connexin 30 and connexin 43. And within the SCN of the mouse, it was found that expression of these specific connexin proteins are time of day dependent, being higher during the day than they are during the night. So moving out of the SCN and back into the hippocampus, we wondered if the hippocampus mimics these day-night changes as well. So on the right here, we have the IHC fluorescent image where we see the connexins in green, and they seem to well approximate the astrocyte processes that are labeled in red. However, when we look at two specific time points in our 24-hour cycle, ZT2 being 2 hours after lights on, and ZT14 being 2 hours after our lights turn off, we don't see a difference in this expression via Western blot. However, this is just a preliminary set of this time course, and with my undergrad, we're currently working on expanding this course to better understand the changes around the clock. So although we didn't find bulk expression changes in connexin 43 during these two time points, with these two specific time points, our lab has found that there are significant changes in the coupling state of these astrocytes. So by using a whole cell patch clamp, we were able to inject sulfurrhodamine B dye, which is small enough to fit through the gap junctions, and you can trace the diffusion of this dye over time. And so what was found is that in the dentate gyrus, the number of coupled astrocytes at night, which is the active phase in the rat, is significantly greater than it is in the early day. And so moving forward with this, and to help further elucidate the mechanism, we want to know, how does gap junction inhibitors affect the coupling of the astrocytes? Do we see the magnitude of this effect equal between time points, or would we see a higher effect at night when there are more astrocytes coupled together than there are during the day? Moving forward, I'm looking to combine not only the techniques that we have in our lab already, but those that I have gained through my time in the MBM program. So this summer, I had the opportunity to complete an external research experience at the University of Iowa with Dr. Ted Abel. And during my rotation, I focused on learning techniques involving sleep deprivation, long-term potentiation, and fear conditioning. And it was really the sleep deprivation that I found most interesting, as it has large implications within the hippocampus itself. And so Dr. Abel's lab has a wealth of publications involving sleep deprivation, but just a few of their major findings relating to how sleep deprivation negatively impacts the hippocampus is that they found that it reduces memory consolidation after fear conditioning. Sleep deprivation decreases dendritic spine density, and it also impairs specific forms of long-term potentiation. So with these neuronal changes that we see within the hippocampus following sleep deprivation, the next question that I hope to ask is, how does sleep deprivation affect the coupling of astrocytes? Is this simply a circadian-based change that we see day to night, or would sleep deprivation cause a decoupling of astrocytes as well? So I'm very excited to get into answering this question very soon. And so with that, I would just like to acknowledge my advisor, Dr. Martha Gillette, as well as all of our Gillette lab members and the undergraduates who have been instrumental to helping me complete these projects. And of course, the last 2 years that I've spent with the miniature brain machinery program, this research would not be possible, and these collaborations would not be possible without them. So thank you.
Ready? Go ahead. So, does dysfunctional gap junction coupling being reported in diseases that cause memory loss, such as Alzheimer's disease?
I don't have specific knowledge of decoupling in Alzheimer's, but a paper was recently published that decoupling of astrocytes does indeed affect learning and memory. But that was just within a conditional knockout model, not in a disease state model.
I'm asking this question because, you know, the ApoE4 is a major genetic risk factor of Alzheimer's disease, and the major factory of ApoE4 is astrocytes. So I wonder whether gap junction coupling affect ApoE4 production and release from the astrocyte that would affect neuronal function.
Yeah, I think that would definitely something interesting to look into, thinking within those networks of astrocytes, if those ApoE4 receptors and the release of that is spread, or if some coupling changes raise or lower that.
So we've looked at intrinsically photosensitive retinal ganglion cells in the retina. And those are the cells that sense the diurnal variations of light that may project to the SCN. My question is whether or not are there other ways of influencing these changes just from like the visual system or some visual activity, and that may affect kind of the changes in the hippocampus. So are there other routes that maybe would affect those changes in the hippocampus through like the visual system, their cortex, and other like experiences during the day besides coming from the SCN?
That's a very interesting question. Specifically we're mainly going off of the Zeitgeber of light. We do have experiments within the lab to where we look at chemicals that can promote phase shifts as well. So there can be chemical manipulation.